Our lecture today is a continuation of the discussion of steroidogenesis. We'll complete the discussion of steroid hormone synthesis in the adrenal cortex, and then discuss the male and female gonadal steroid synthesis. Switch over to the computer now, and just briefly recall that we had ended the previous lecture on this information talking about the CAMP, the cyclic AMP dependent level of steroidogenic gene expression, discussing the acute and the chronic types, the temporally based distinction between these two, obviously on the scale of minutes to hours. Now we want to talk about the one gene expression dependent aspect of both of these, assuming, of course, that acute leads into chronic after the minutes are over and the hours kick in. In other words, there is one gene that must be expressed in order for the adrenal steroid, and this is true as well for the gonadal steroids synthesis to be initiated and to be sustained. And this is dependent upon this STAR, the star of our show, as we had mentioned earlier. And that acronym stands for Steroidogenic Acute Regulatory Protein. So the acute regulatory is key. It is acutely regulated, which makes us think immediately of turnover rate. It probably has a very high turnover rate in that this gene must be induced and, of course, expressed all the way to the production of the functional gene product. One of the research articles that I referred you to in the first part of this lecture discusses the post-translational processing of STAR. There is a mitochondrial binding domain on this protein that is revealed upon post-translational processing, and of course thereby it is site-directed to the mitochondria. And it is here that this STAR protein produces its function. So in attempting to stimulate cyclic AMP dependent steroid synthesis within the adrenal cortex, it is found that the process is cyclohexamide sensitive. Cyclohexamide is a protein synthesis inhibitor. It blocks at the level of translation. We'll talk about another molecule in later lectures on the HPA that blocks transcription. Cyclohexamide blocks protein synthesis at the level of translation. Therefore, if this aspect of steroidogenesis, ligand transduced steroidogenesis, is cyclohexamide sensitive, then we conclude that it is protein synthesis dependent. So the process depends on the synthesis of a new protein, a de novo synthesis of a protein, where if it is present at constitutive levels, they are insufficient to mediate the receptor cyclic AMP dependent action that we just described. So this is required for the cholesterol delivery to the inner mitochondrial membrane. As you recall in the prior discussions as well, we talked about another aspect of the cyclic AMP dependent steroidogenesis in the adrenal cortical tissue as well as the gonadal steroid synthesizing cells, and that is the cyclic AMP dependent, pKa dependent activation of cholesterol ester hydrolase enzyme that will break down the stored cholesterol form and make it free and available within the cytoplasm for this protein STAR to bind it and bring about the transport into the mitochondria, and therefore we see its key role in getting the cholesterol from the cytoplasm ultimately to the through the inner mitochondrial membrane, where the P450 11A1 enzyme can convert it to pregnenolone, that first rate-limiting step of the actual steroid synthesis process. So there is a rapid increase in the acute response to the ACTH cyclic AMP event. So therefore, the ligand transduction of cyclic AMP synthesis within the target cells, I should say. So this illustration takes us through that, as well as a few other things, the non-cyclic AMP dependent, and this is from the molecular endocrinology article that I had referred you to. So our focus here is on, you notice they have the LH and ACTH, so it's a common event at both types of steroidogenic tissue. We're focusing now on the ACTH-mediated signal transduction at the G-coupled protein receptor in the membrane of the target cell, focusing on the activation of adenylate cyclase to produce cyclic AMP from ATP. So we are focusing on this in the cyclic AMP-dependent pathway, getting to the STAR gene. And this is the focus of our slide, the STAR gene with its promoter region shown here to induce the transcription or modulate it positively, so there's an upregulation such that there's more STAR message, more translation, more STAR to be phosphorylated and become activated to bind to the mitochondrial protein and then transport the cholesterol as it becomes available. So in this cyclic AMP dependent pathway, we're missing one step that is not shown on this illustration, of course, and that is the pKa activation of the cholesterol ester hydrolase that frees up the cholesterol to be transported by the STAR. So slides like this are always quite confusing, but we had mentioned cyclic AMP dependent and cyclic AMP independent mechanisms for affecting steroidogenesis in tissues. Here we're seeing that many of those, including the chloride, the calcium, the growth factors, and even some of the immune system like arachidonic acid mediated pathways as shown here that are also related to cyclic AMP events but independent of the pKa, all leading to arachidonic acid metabolites that can have an inhibitory effect on the transcription of STAR gene, and then through the growth hormone receptors working through a variety of pathways having some effects on the transcription of that gene as well. So we're not focusing so much on these alternate pathways in here as we are the cyclic AMP dependent, pKa dependent transcription of the STAR gene that is essential to trigger the steroidogenic pathway, primarily by providing the initial substrate. So this slide is summarizing some of the cyclic AMP dependent, pKa dependent pathways that are still somewhat uncertain for affecting the various genes other than STAR. STAR is shown here on the far right, and this other one, the ADX that we had mentioned. But we recognize these, the CYP11A1. This is the key enzyme for the conversion of the cholesterol to the pregnenolone. These two genes have gene products that are key for differentiating between the aldosterone synthesis versus the androstenedione or the androgen estrogen hormones in the zona reticularis. And the CYP17 is a key one in the final steroidogenic production of the glucocorticoids, and we'll take another look at that in a moment a bit more closely. So many of these transcription factors, the SF1 we mentioned, the SP1 we mentioned or will mention, the ATF2, the PSF, P45, NRB, ASP, SP3, we haven't had opportunities to discuss those in any great detail in this class. They're pretty much beyond the scope of our discussion or focus on transcription factors, but it certainly reminds us that there's a tremendous complexity of interaction of transcription factors in the regulation of gene expression. So rather than try to remove them from the slide to simplify it, I leave it to remind us of the low level of complexity that we're trying to grasp and gain and further our appreciation of regulated gene expression. So the CYP, it is CYP, did I type that in wrong? Yeah, sorry, I typed that in wrong. CYP11A1 gene encodes for the mitochondrial enzyme P45011A1, also called P450-SCC, as we had said. That's the key, critical one for converting the cholesterol to the pregnenolone and initiating that first cascade down the biosynthetic pathway. We noted that the SF1 binding is required at 2 sites in general, but now we note at 2 sites on the promoter region at the minus 40 base pairs upstream from the start site and minus 1600 base pairs upstream. So with our discussion of promoter analysis, in our mind we should immediately be imagining the studies using a reporter gene where they're either truncating or have used substitution or deletion types approaches to identify where these SF1 dependent regions for the expression of this gene are located. It is also potentiated by C-Jun. Again, another emphasis of the complexity of the interaction between transcription factors. So we did talk about C-Jun. It's one of the immediate early genes, the C-Phos and the C-Jun, and they target primarily the AP1 consensus sequence. So somehow when the C-Jun is expressed in these cells, it'll potentiate the SF1 mediated increased expression of the CYP11A1 gene, producing more of the P450-SCC enzyme to promote the steroidogenesis. So this is a very different way of thinking about hormone synthesis. The peptide hormone synthesis, we merely had to increase the expression of that peptide hormone gene, get more of it into the RER, more of it into the Golgi, more of it into more vesicles, and you had more to be released. Now we have to keep in mind all of these enzymes in these biosynthetic, biochemical pathways, and we're expressing the genes to get more of these enzymes, gene products, into a pool that can be activated and to replace the enzymes that are consumed somehow. All this need for increased gene expression makes us think that the turnover time, the half-life for these synthetic enzymes is relatively short as well. Otherwise, we wouldn't have such a dependency on sustaining the gene expression, specifically in our mind now thinking of the ACTH receptor activation. So with the rate-limiting enzyme, you want to have plenty of that around. So otherwise, this steroidogenic process is dependent on the 2 steps that we talked about before this key step here, the rate-limiting step, and that was the activation, the receptor-dependent activation of the cholesterol ester hydrolase, and the synthesis, increased expression of the gene for STAR. So you have to have enough cholesterol. If you don't have enough precursor, it doesn't matter how much of the the P450-11A1 you have and have activated. If the precursor isn't there, there's nothing for it to act on enzymatically. So there's a key interplay or a convergence from the receptor onto those two key steps to begin with, as well as on the expression of the key enzyme. So another aspect of the expression of the CYP, I kept putting CPY in there, 11A gene, is that there's a novel zinc finger protein that has been discovered. And I use this more to remind us of the zinc finger proteins than another transcription factor to throw in our list. It is a positive regulator as well. And as well, there's the ubiquitously expressed transcription factor, the SP1, that regulates the cyclic AMP-dependent transcription, and this is for the human, based upon studies of human adrenocortical cells. So the SF1 and the SP1 act cooperatively in the bovine adrenocortical cells, and that SF1-SP1 cooperative interaction, is enhanced by the presence of a CBP, and we'll talk about that in a moment. So basically a Krebs binding protein. So the number of proteins, transcription factors, and co-activators that begin to stick together increases and demonstrates a greater level of complexity as well. So now we can talk a little bit about the recruitment of co-activators and co-repressors, and there is a major role by STAR in that the co-activators, the proteins required to regulate, the proteins recruited to these regulatory regions of the DNA bound by the transcription factors will become located on this STAR promoter region. So the steroid receptor co-activator, the SRC1, the Krebs binding protein, the one that we mentioned on the previous slide, P300. Again, we're beginning to see more and more that we are not going to have opportunities to learn any details about, but certainly we should be able to recognize them and associate them with the recruitment of co-activators or co-repressors. So on the STAR promoter region, these co-activator types of proteins are essential for bringing about the STAR gene expression. And what these do is they possess an intrinsic histone acetyltransferase activity. So this we have discussed before, and what happens is that intrinsic enzymatic activity brings about the acetylation of histone proteins. We know those are the proteins that help to coil the DNA into the nucleosomes at the basic level. So acetylation of the histone proteins increases the accessibility of the transcription factors to the nucleosomal DNA and correlates with transcriptional activity. So to interpret this statement, when the histones are acetylated, then they decrease their affinity for the DNA, and then you can have additional uncoiling of the DNA from the nucleosome, relaxing that level of the coiling of the DNA. Of course, that just creates opportunities for other transcription factors to loosen up the double helix so that the start site is more available for the transcription factor complexes, including the RNA polymerase, to bind. And you see that it correlates with transcriptional activity. We would predict that this is a positive correlation. The more you loosen the DNA, the more you increase the statistical probability that RNA transcriptase is going to be able to get in there and bind. Co-repressors, on the other hand, for example, this MSyn3A, mediates the inhibition of this the CYP17 gene expression and recruits a histone deacetylase. So already, if you deacetylate or if you acetylate histones and you relax it, we can predict that if you deacetylate, you tighten it, and you bring about the repression. So there's this SF1 NONO-PSF complex that binds the promoter and is associated with the deacetylation and tightening of the nucleosomal coil on this CYP17 gene in particular, and reduces the expression. So this is summarized in this illustration. So the ACTH coming ultimately to the activation of adenylate cyclase, the transduction through its receptor, to the cyclic AMP, to the PKA, and there's this MKP that becomes phosphorylated and then has this effect on this complex, a type of MAP kinase that is responsible for the basal phosphorylation. And we see that in the basal phosphorylated state, the SF1 with these various transcription factors and co-repressors, the MSyn3A was a co-repressor, has the histone deacetylase activity. And when this phosphate group is removed in this process, coming to the dephosphorylated SF1, we see that the intrinsic acetylation, histone acetylation activity can occur, and you increase the transcription rate of the CYP17 gene, leading to the cortisol and aldosterone pathways. So this is an essential aspect of ACTH cyclic AMP dependent gene expression of one of the other biosynthetic enzymes versus what we've discussed already previously, this CYP17, bringing about a key enzyme in the production specifically of cortisol and aldosterone as the final steroidal products of the biosynthetic pathway. Excuse me. So remember, what we've said is that there has to be key enzymes at the end of certain biosynthetic pathways so that the product of that enzymatic reaction isn't merely an intermediate for the next enzyme, and it builds up to a high enough level where it has the opportunity to diffuse away, and that's secretion. So the mechanism of secretion for the steroid hormones is free diffusion from the point of synthesis, which we predict to be the point of highest concentration. And remember that those carrier proteins in the plasma are really critical because they occupy or create an environment that favors the location of the steroid hormones in the plasma, so that they reach a higher concentration and they're drawn away, and sustains that concentration gradient from the original source, that final synthetic enzyme, to the final point of delivery, in this case for secretion into the plasma. Well, now we can transition over to the sex steroid synthesis with this similar illustration as we saw in the previous slide for the adrenal cortex. And again, we note key genes to be expressed in these sex steroid synthesizing cells. We recognize STAR. We already noted that it is a key player in this process as well. The CYP11A, so that first key enzyme, we note that STAR gene product is targeted to the mitochondrion, this little box up here, as is the gene product of the CYP11A, the P450 SCC. And those, of course, are key factors in getting the cholesterol into the mitochondrion so it can be converted to the pregnenolone. So that's a common original part of the process. So if we could picture the gonadotropin receptor here somewhere, of course, it would have to branch into the pathway for increasing the gene expression of the star, as well on over to any enzymes involved in providing the cholesterol that's necessary. And for the sex steroid synthesis, except for the corpus luteum, I provided you that one reference for the review on progesterone synthesis in the corpus luteum yellow, because of the tremendous cholesterol ester reserves within it. But other than that, the steroid synthesizing cells, the ones that originate this process, either synthesize their own cholesterol or take it up from the interstitial space, interstitial fluid, as either LDLs or HDLs. And that's kind of interesting reading, but about which one uses which type for which type of synthesis, and I can leave that for your own perusal in those review articles that I've recommended. But the one distinction between the adrenal cortex and the steroidogenic cells, exclusive of the corpus luteum, is the source of the cholesterol. But still, there is the neutral cholesterol ester hydrolase. That gene is expressed in these cells, and it is present. So there are some stored lipids in the cytoplasm upon which that enzyme can act to free up cholesterol. So just a variation on the potential sources of the cholesterol for the sex steroid synthesis. It's not strictly dependent upon the cholesterol ester hydrolase activation, but it's still a key component. So this gets us to the pregnenolone in virtually the same fashion, but slightly different on the aspect of the sources of cholesterol. And then the pathways are divergent and convergent, as you can see, until ultimately testosterone and estradiol are produced. We note that estradiol can be synthesized from two sources: estrone through the 17-beta HSD enzyme that acts at other locations as well. And here we see the P450 aromatase that can not only act on the conversion of the androstenedione to estrone, but also, as we recall, the aromatase is essential for the conversion of testosterone to estradiol, and this is the 17-beta estradiol. So here's that CYP17. If we follow it out to here, we see that it can act on various levels of the biosynthetic process of relevance. Here we see that it takes it to the androstenedione, which was essential in the adrenal cortex, the zona reticulata, ultimately to get to the adrenal cortical synthesis of the gonadal steroids. So there is a common type of array of gene expression in the adrenal cortical zona reticulata cells because they do have the capability to produce the androgens and the estrogens. Well, now we transfer to slightly more simplified illustrations on all these biosynthetic pathways to begin to discuss this two-cell, two-hormone model for the synthesis of the steroids. And the first one we look at is in the male, and we challenge this idea of the two gonadotropins, two cells for the synthesis of specifically the androgens in the male. But we still see that there's two cells, two gonadotropins, but two different sex steroids in this biosynthetic pathway for males to accomplish the two goals, spermatogenesis or gametogenesis in general, and the support, the development, support, and sustenance of the reproductive organs, and of course we know contributing to secondary sexual characteristics as well. So first we take note of this illustration of pregnenolone. It shows us the labeling of the carbon molecules. Here's 1 through 9 there. There's 10, 11, 12, 13, 14, 15, 16, 17, 18. We notice that 18 and 19 are not within the rings, and then 20 and 21 are the final carbons labeled here in this side chain, and then there's rings A, B, C, and D. So we note over here the estradiol. Of course, these two carbon groups are gone, 20 and 21 are gone, and they're replaced by a hydroxyl group. As well over here, the hydroxyl group is maintained on carbon number 3, making it, of course, two OH groups, which are alcohols, so estradiol, and this is carbon number 17, so it's 17 beta formation for this bond, 17 beta estradiol. So that's kind of the organic chemistry for the estradiol. And remember, this ring is aromatized by the aromatase either from estrone or testosterone. So let's dig into the three-gland, three-hormone process here model. Gland 1, the hypothalamus, hormone 1, the GnRH, targeting the anterior pituitary gland, the gonadotropes for LH and FSH secretion and synthesis. The FSH, let's approach it this way. The Sertoli cells have a high concentration of FSH receptors and not LH receptors. Therefore, when it finally gets into the male gonad, it will bind to the receptor on the Sertoli cell and stimulate the synthesis and secretion of inhibin that has a specific negative feedback at the anterior pituitary selectively on the FSH, synthesis and secretion. Also, the FSH promotes the androgen binding protein gene expression, so sustains that. Also, as we will see, the expression of the aromatase gene for the conversion of testosterone to estradiol within the Sertoli cell. So the FSH is not essential for the production of testosterone in the male gonad. LH is. So the Leydig cell, the interstitial cells of Leydig, are responsible, or express high levels of LH gene, and therefore the LH, when it gets there, will bind there and trigger the enzymatic process, as we'll describe, through the cyclic AMP-dependent gene expression of the enzymes and activation of enzymes, ultimately to produce testosterone. Testosterone can be converted to DHT, which also contributes directly to the development of sperm cells. But the testosterone can diffuse into the Sertoli cell and is held there by the androgen binding protein. Aromatase acts on it and converts it to estradiol, and the Sertoli cells actually have estrogen receptors and EREs on the significant genes for sustaining the capability of the Sertoli cells to promote spermatogenesis. So an aromatase inhibitor would block this step and inhibit sperm cell production. The Leydig cells are the ones that produce the testosterone. We see it here. There's a carboxy. No, is that what it is? It's an one anyway. This is a ketone here, so it's testosterone, not the OL for the estradiol when aromatase converts it. So testosterone is shown here in its chemical format. So the testosterone is not responsible for the negative feedback on the hypothalamus and the anterior pituitary. The expression of the aromatase gene is found in the target cells, responsible for those two negative feedbacks. We talked about that earlier when we were discussing negative feedback in general. So the testosterone in the male is important for targeting the duct system, the accessory glands, the secondary organs, and of course either on its own or as dihydrotestosterone for the development and maintenance of secondary sexual characteristics. Now let's take a look at this in a bit more detail. So the LH acts at various enzymatic sites within the Leydig cells to prompt the synthesis of testosterone. So these slides emphasize the cyclic AMP-dependent activation of this hydrolase here. We now know that that is cholesterol ester hydrolase, and it converts the cholesterol ester to cholesterol. Star brings it to the mitochondria. At the mitochondria, there's a mitochondrial transport side chain cleavage that occurs. This is the P450 11A1 or SCC that converts the cholesterol to the pregnenolone. There are additional hydroxylases that convert it to hydroxypregnenolone, then dehydroepiandrosterone, then androstenedione, and finally to testosterone. So I'm not asking you to remember all the enzymes, but I certainly hope you can recognize these names, maybe not necessarily in their sequence, but that any of these are between pregnenolone and testosterone, and thereby, of course, between pregnenolone and estradiol. It's important to remember the term estradenedione because we'll see that that's critical for the two-cell, two-gonadotropin model in the female. So this slide is showing us again that there is two-cell, two-gonadotropin mechanism for testicular steroidogenesis, not just for androgens, and that's because, of course, estrogen is also produced in the male gonad, and it's the testosterone that diffuses over to the Sertoli cell, and the aromatase gene and enzyme activity is sustained by the FSH receptor through the cyclic AMP, so that estradiol is produced. So the latent hydrolase, the cholesterol ester hydrolase, is activated, mobilizes the cholesterol from the cholesterol esters, the transport of the cholesterol into the mitochondria by Star, and the synthesis of the mitochondrial C27 and microsomal C21 side chain cleavage enzymes those enzymes are induced. These would be the CYP 27 and 21 that are also important in addition to the 11A1. And the delta 5 pathway steroidogenesis prevails in the male, and that is by bringing it to the synthesis of testosterone. Now, once testosterone is synthesized, we remind ourselves that there is the degradation end of this. So here is cholesterol. We see that it has the ABCD rings, but additional carbons up above. So those are cleaved by the P450 11A1 enzyme to make the pregnenolone, as you recall. So this cholesterol in the Leydig cells, it's primarily the delta 5 pathway that brings it to the production of testosterone. The dihydrotestosterone can be produced, and it is shown here, the 5-alpha dihydrotestosterone. And then, of course, the Sertoli cells in the brain and the anterior pituitary can convert that to the estradiol. So we note the distinction between testosterone and 5-alpha dihydrotestosterone. We lose the double bond within the A ring here. Further, the testosterone is, now this is the changing of testosterone. I guess we would still, I mistakenly called it catabolic. This is still anabolic metabolism of testosterone because these are biologically active. The catabolism after the 5-alpha reductase down that pathway to produce the 5-alpha DHT is enzymatic changes to produce these byproducts. These would be detected in the urine predominantly, and the aromatase bringing it to the estradiol that are converted to the catechol estrogens, and these would be the molecules contained primarily in the urine. So be careful not to confuse any of these molecular structures with the ones that I've described more specifically and would hope you can recognize. Now let's spend just a bit of time on the HPG in the female for the 2-cell 2-gonadotropin model. We first recall that the follicles develop to the stage, especially the mature follicle shown here, where the follicular cells develop into the theca externa and theca interna. It's the theca interna cells that we're primarily going to focus on. Note the rich capillary bed in that area for delivery and washing away of hormones. And as well, within the follicle, there are the granulosa cells, a collection of cells here called the membrana granulosa in the mature follicle, where it ultimately becomes important. But originally, all the developing follicles, remember in the pre-ovulatory or the follicular phase, you have recruitment and development of follicles. In humans, one of those becomes the mature follicle, but along the way, they all contribute to synthesizing estrogen and some progesterone. And it's an interaction between the theca internal cells and the granulosa cells for the 2-cell part of the 2-cell 2-gonadotropin model of female gonadal steroid synthesis. After ovulation, luteinization of the granulosa and some of the thecal cells occurs. This is described in, again, the review article on cholesterol uptake and progesterone synthesis in the corpus luteum. We're not going to get into details, but again, it's pretty interesting reading. So there is a process, initial process just after ovulation called luteinization, where these cells undergo a significant differentiation, and it's really quite quick. Sometimes within 30 minutes of ovulation, you can have this transition to a high level of progesterone synthesis. And then after luteinization, you can see this large corpus luteum here. We can't really appreciate the yellowish color, but tremendous deposits of cholesterol ester in the luteal and granulosa cells that have differentiated into corpus luteal cells now. So in this model then, what we are seeing again is the gland 1, hormone 1, gland 1, hormones 2. In this case, the LH is targeting the thecal cells because they selectively express the LH receptor gene. The granulosa cells selectively express the FSH gene and are targeted by FSH, and they are the cells of origin of the inhibin that produce the selective negative feedback at the anterior pituitary on FSH. So here we also see that the LH can contribute to corpus luteal progesterone synthesis, although when you read that review article, you'll see that this is a contributor but not the main role for progesterone synthesis. It is somewhat self-sustaining once luteinization has finished. So here we see the progesterone. These 2 carbon molecules persist here. It's the double bond O over here. So it is a ketone, therefore progesterone. So what we will see is that the thecal cells will convert cholesterol to the androstenedione, and then the rest is left up to the granulosa cells. Of course, the estradiol is important for tubular organ development and sustenance and changes through the reproductive cycle. In some animals, mating produces sensory signals so that there's a neural feedback that is positive for bringing about the LH surge in the estrous cycle for inducible ovulators. But we're showing the estradiol here producing a positive feedback at the anterior pituitary, positive or negative at the hypothalamus, and the progesterone being involved in the negative feedback at the level of the hypothalamus for the female reproductive HPG axis. So it is more complex. As you recall, in the pre-ovulatory phase, the follicular phase, the estradiol levels are increasing as the follicles are recruited and are developing and trigger a positive feedback so that you get the LH surge. There's also a smaller FSH surge that accompanies that. So estradiol has positive feedback during that portion of the menstrual or estrous cycle. In this slide, we're showing the information for the inducible ovulators. So still, and this is in estrus cycling animals, the follicular growth is going to occur in the follicular phase. Estradiol will be produced. This brings about estrus, and then there will not be ovulation in these animals. No LH surge will be produced unless there is mating. So the cervical stimulation provides a sensory stimulus through the spinal cord and through acetylcholine and norepinephrine in the preoptic area of the hypothalamus. You get a GnRH surge that triggers the LH surge at the level of the anterior pituitary, and then there will be ovulation. And as we know in rabbits, not only are they inducible ovulators, they also have multiple ovulations. Again, then, we see that in the thecal cells for the two-gonadotropin model, the pathway is the same for the LH-stimulated enzymatic synthesis, but the genes are expressed only up to the level where the enzyme responsible for the production of androstenedione occurs. So this builds up and diffuses away, reaches the granulosa cells nearby, and in the granulosa cells, the enzymes are expressed to convert, the enzyme genes are expressed to convert that to testosterone, as well as the aromatase gene, and the aromatase gene expression, as well as the others most likely, and activation is sustained by the cyclic AMP produced through the FSH binding to its receptor, and that is what ultimately produces the estradiol in the female gonads. This slide was discussed in an earlier lecture on the menstrual and estrous cycle. I'll refer you back to that time point, and you can read it and see what's going on. Let's just quickly focus on the key role of cyclic AMP in association with high levels of estrogen, as well as the LH surge within 24 hours of ovulation. And this cyclic AMP in the anterior pituitary is sustained and, of course, associated with the prolonged release of the LH and the ultimate effect on ovulation. I believe this also was slide was also included in a previous lecture. I will expect you to be able to differentiate between ethinyl estradiol. Here we see the ethinyl, of course, is referring to the triple bond here on the carbon 17. So it's not 17 beta estradiol. It still is estradiol, but there is an ethinyl group also attached there. And then norethadrone is a modification of the progesterone with the ethinyl group located there as well. So notice the difference, the C double bond O for the ON, similar to the progesterone. So these are modified estradiol and progesterone molecules that, of course, are orally active steroids used in some of the birth control pills. So this ends our discussion of steroidogenesis. I appreciate your time and attention, especially somebody out there is turning my pip off, and I keep turning it back on. We are now just one set of lectures away from the test 4 to discuss the hypothalamic-pituitary adrenal axis. Of course, this lecture on steroidogenesis covered gland 3, hormones 3, as well as the receptors for hormones 2 quite extensively. So the HPG axis will be the last part of the lectures in this class, focusing primarily on hormone 1, gland 1, hormone 1, gland 2, hormone 2, and then that will be all covered for test 4, but the menstrual cycle and the estrous cycle and the HPG axes in general, of course, are on the test 4, and the HPT and the HPA axis with the focus on the steroidogenesis for both the HPA and the HPG axis on this test 4. So, hope you all do well in the upcoming test.